Hello students, I am Dr. Manisha Girase, Professor of Microbiology. Today we are going to study paper Fermentation Technology. Code of paper is MB365. In this paper we are going to study today's topic is polio vaccine production. In our syllabus we have to study vaccine production as well as anti-serum production. In today's lecture we will cover polio vaccine production. Vaccines are nothing but the biological preparations. These vaccines provide active acquired type of immunity. Now active immunity means body is actively involved in this type of immunity, means body synthesize various compounds which are necessary for development of antibody. As it is acquired due to the preparation, therefore it is known as acquired. This type of immunity is developed against many infectious diseases. Now in these vaccines there are different agents that resemble to the disease causing organism, that is pathogen. Means in our body we take certain preparation which looks like or which resembles the pathogen. Now these preparations are prepared from various things like weakened organism or sometimes killed organism. It is also prepared from the toxin, means the toxin of the organism that is converted into non-toxic form. This non-toxic form is known as toxoid. And many times rather than using killed or live organism, there is use of a part of the microorganism. For example, for preparation of hepatitis B vaccine, there is use of surface antigen. So likewise, different preparations are used. Now according to this, vaccines are of different types. Some vaccines are live vaccines, means they contain live bacteria. Some vaccines are known as dead vaccines, means they contain dead bacteria or virus. Some are subunit vaccines, means rather than the whole organism, a part of the organism is used, and some are genetically engineered, means that part of the vaccine that is synthesized by the organism using genetic engineering technique. Now vaccines are administered and in the body to develop the immunity. This is the most effective method of preventing infectious diseases. And all of us are aware how by using vaccines we have prevented COVID disease. Now these vaccines help us in eradication of diseases. For example, there was a widespread of smallpox, and this widespread immunity amongst the population that was developed for the disease smallpox. Now this smallpox disease is completely eradicated. Now some diseases are restricted, means they are controlled, like polio vaccines is now in control due to vaccination, measles, tetanus. All these diseases, they are controlled or they are restricted due to the use of vaccines. Therefore, there is a massive global vaccination campaign for various diseases. Now for polio disease, from past 20 years, there is massive vaccination, and because of this, now this disease is controlled in many parts like in Africa and Asia, it is under control. Polio vaccine is against the disease polio, or it is also known as poliomyelitis. This disease is caused by polio virus. Now this virus infects the spinal cord of the human being, and as well as it infects the brain. And because of this infection to the spinal cord and brain, there is development of paralysis, and sometimes there may be even death of the patient. Generally, children who are below 5 years of age, they are most susceptible to this disease. This is the structure of polio virus. It is RNA virus. It is having glycoprotein. It is having capsid, and it is having a membrane, which is membranous organism virus is there. Now this polio virus was, for vaccine we have to grow the organism, and first time the polio virus is grown using tissue culture technique in 1949. Polio disease is caused by 3 different serotypes of the polio. They are isolated and identified in 1951. Now, first trial of the Salk vaccine means it is a kill type of vaccine. It is performed in 1954, and the first use of Sabin vaccine, it is a attenuated organism. It is developed in 1958. Now, mechanism of polio vaccine means when virus particle either inactivated or it is attenuated when it enters in the body. Major histocompatibility to complex antigen presenting cells, they recognize this. Then there is activation, there is formation of cytotoxic cells, there is formation of memory cells, as well as there is activation of B cells. These B cells activated by the viral antigen, they produce antibodies and memory B cells, while there is production of memory T cells and cytotoxic cells. This is how the vaccine work, means it enters in a body in the same way as the pathogen enters, virus particle enter, and same way it activates the energy, but there is no entry of the infectious organism, only the inactivated or killed organism is entering in the body, and still it is developing the formation of cytotoxic cells or antibodies against this virus, and this is how they protect us from the infections. Now, there are two general types of polio vaccine. One is inactivated polio vaccine, that is known as IPV. This inactivated polio vaccine is known as Salk vaccine because it is developed by the scientist Salk. And oral polio vaccine, that is known as Sabin vaccine, and this is given orally. It contains attenuated, means these organisms are the live organism, but only they have lost their pathogenicity. Now, in both the types of vaccine, either it is Salk vaccine or Sabin vaccine, there is use of all three strains of the vaccine, all three serotypes of the vaccine. Now, in the process of production of the Salk vaccine, which is a inactivated or kill type of vaccine, first there is growth of the virus. Now, we are all aware that virus require living media for their growth. They cannot be cultivated on inactivated media, non-living media. So, these polio vaccine, for production of polio vaccine, polio virus is cultivated on human diploid cell culture. All the standard virulent, all three serotypes, they are inoculated in the separate cell culture. They are incubated till the sufficient titer, means sufficient number of virus particles are formed. Now, in this cell, inside the cell, virus particle will grow and that fluid will contain virus particle. Then this fluid is collected and it is filtered, and this clarified medium will contain the virus particle. Now, after this cultivation, these virus particles are inactivated by using various chemicals like dilute formalin or beta propionolactone, and after this, as it is in very dilute form, further step is concentration of virus. For concentration of virus, adsorption mechanism is used, and then after adsorbed virus particle, they are eluted. And this step is repeated for all three serotypes, and finally all three serotypes are mixed together, means there is blending of the virus particles. Now, in the similar way, oral polio vaccine is used, that is the Sabin type of vaccine. Now, in this vaccine, virus particle is not killed. The non-pathogenic strain of this virus is used, means it is known as attenuated, means this virus particle will grow inside the body, multiply inside the body, only thing it will not produce the disease. In the same way, all three serotypes are used. This is developed by scientist Albert Sabin. It is live attenuated viral vaccine. Now, this virus particle, when it enters in the body, it will produce a local gastrointestinal infection, but that will be very mild type of infection. It is a live vaccine, therefore, one should not inject this, because in injection killed vaccine is used, and oral vaccines they are attenuated vaccine, they should not be injected. So, Salk vaccine is given in the form of injection, and Sabin vaccine is given in the form of oral drops. Now all three serotypes after growth, they are now how they are attenuated. Now rather than cultivating them in the human cells, they are cultivated in the monkey kidney cells. Monkey kidney cells are not their usual host. Human being is usual host. If we continuously cultivate this organism in monkey kidney cells, they fail to create infection in human. So continuously they are allowed to grow in the monkey kidney cell. Then this growth in the new host make them less infectious to the human being. All three serotypes, they are attenuated in the same way, and then they are grown in the human diploid cell culture and incubated till sufficient growth is there. Now in this cell culture, there is the fluid will contain organism, as well as due to death of the cells, debris will be formed. Therefore these debris are separated by means of filtration. Then whatever clear fluid is obtained, that is used, it will contain viruses. Then further step is concentration. Again this is done by same way, adsorption. Then after adsorption, it is eluted, and then blending of all three serotypes. Now this attenuated virus, it will not create infection, but it will retain or it will maintain its antigenicity. Means it can stimulate antigenic response in the body, so that there will be formation of cytotoxic cells and antibody. In this vaccine, there is use of stabilizers like sucrose or trehalose. These sugars are used, and there can be a use of arginine hydrochloride as a stabilizing agent. Now after the production, the next step is filling and drying, because it should reach to the patient. For this there is filling process is there. Now some vaccines are packed in the terms of bulk volume. So the bulk vaccine is distributed, or sometimes there is use of a single dose. So bulk vaccine is packed in terms of vials. So vial will contain a dose for many patients, and ampule will contain a single dose. So it can be used for one person. So these ampules are sealed and vials are capped with rubber capping. So this is how the filling is done. This is the overall process of vaccine production. As we have seen, the cells viruses are grown. Then after this, there is removal of debris by means of filtration. Then there is inactivation step, then concentration step. Then further they are purified by means of chromatographic technique. Then there is concentration, because it is in a very diluted form. Then all three forms are mixed together, that is known as blending. And the last step is packaging. Now after the production, the last step in the industry is quality control, because as it is after production, it cannot be marketed unless and until we are sure about its quality. Quality we have to check in terms of safety as well as efficacy. Efficacy means its efficiency, whether it is efficient in providing immunity or not, that we have to check. And the safety means whether it is safe or not, whether it contains any other material which is not safe or which is harmful for the patient. So for every batch we have to check efficacy and safety test. So these efficacy and safety test, that is known as quality control, and these quality control are done by two ways. One is in process control, means as the process is going on, at every step we have to check its quality, and this is known as in process control. And next is final product control, means once final product is formed, we have to check whether the final product is of good quality or not. Means it is not a one step quality control. During the process, whole process, we have to control the process, and a final product should be also checked for its quality. So in process control is when virus particles are formed, we have to titrate before they are inactivated, means how much number of virus particles are present, that is the titration is done before inactivation of the virus. Then whether virus is having adequate infectivity of the virus from the culture, it indicates that it is not inactivated properly if it is showing infectivity. So a rigorous examination of tissue culture we have to check, because in tissues or cells, there may be some contamination. So we have to exclude, we have to remove this contamination from the human diploid cell which we are using for the cultivation of the virus. Then final product control includes potency test. Now meaning of potency is to check its power. Now this potency testing of the killed vaccine is done by 3+3 Quantal assay. Now why this 3+3 Quantal assay? In this case, there are 3 logarithmic serial doses of the vaccine and compared with the standard vaccine is done. Now what do we mean by logarithmic serial doses? Logarithmic dose means suppose first dose is 1 ml, then its next logarithmic lesser dose can be 0.1 ml and again one logarithmic lesser dose is 0.01 ml. Means it is in the form of 10 raised to 0, 10 raised to -1 and 10 raised to -2. These are the logarithmic scale. In these 3 scales, we have to take our test vaccine which is synthesized, and in the same dose we have to take the standard vaccine, and these 3 logarithmic doses, they are inoculated in the test animal and then their efficiency is checked. Now Salk vaccine, which is a inactivated or killed vaccine, is tested for residual virus if there are any residual viruses which are not inactivated, because this Salk vaccine we are going to inject in a patient, so it should not contain any live virus particle. So for this, the final produced product that is inoculated in the tissue culture, it should not show any histological changes because it doesn't contain any live virus. But if there are any histological changes, then it indicates that it contains some live viruses. So Sabin vaccine, it is a attenuated vaccine. It is tested for reversion, where by chance the non-pathogenic or attenuated form may turn into pathogenic form. So that care we have to check while using Sabin. Means for Salk vaccine we have to check whether there is any live vaccine or not, and for Sabin vaccine we have to check whether there is any reverted pathogenic virus or not, that we have to check, and for this we have to use animal. In spinal cord of the monkey we have to inject the Sabin vaccine and then check whether there are any symptom of the disease or abnormality in the monkey. If no symptoms are found, means there are no reverted virus particles are there. Then the next test is sterility testing, means here we have to check whether the preparation is free from any living form or not. For this, membrane filtration technique is used. Then in this technique we have to check there should not be presence of any aerobic, anaerobic or even fungi should be absent. All forms of the living being should be absent, that checking is done. Also it should be free from abnormal toxicity. So vaccines are checked for toxicity testing. These toxic contaminant, they are checked by using animal guinea pigs. In the guinea pig, if the vaccine, 1 human dose is injected and that guinea pig is observed for any reaction due to the toxic compound, that is confirmed. Then in this preparation we have to check the presence of aluminum and calcium, because this aluminum hydroxide and aluminum phosphate, they are used as adjuvants to increase the antigenicity of the vaccine. So in the vaccine their concentration should not increase more than 1.25 milligram per dose, and calcium should not be more than 1.3 milligram per dose. Formalin is also added for inactivation purpose. It should not be more than 0.02%, and the concentration preservative is phenol. It should not exceed 0.25%. So this is all about the preparation of polio vaccine. I think the preparation of Sabin vaccine and Salk vaccine both are clear to you. Thank you.